Um, what I'm going to tell you something today about is bacteria, infections, and antibiotics. And to get you started is I want to take a look with you at the tree of life. And what you see here at the tree of life is we have bacteria, archaea, which are you could see sort of as a well ancient forms of bacteria, and eukaryotes. And just to give you an idea of how many of those we study in microbiology, all the ones in the big blue circle are bacteria or archaea. Um, these three groups show us the molds, the slimes, the fungi, and several of the parasites. So also these are all considered to be microorganisms. And actually, where we, most of us actually spend our time is only on those two small groups, the animals and the plants. So there's in the tree of life a large amount of organisms, which are all microorganisms, and just as a few other, well, two other groups that we usually study a lot. So today we're going to focus on the other groups. And why do we study microorganisms? Well, I don't know how to say this number, but just to give you a comparison, this was the amount of humans on the planet yesterday. And just to give you order of magnitude, so there's many, many more bacteria on the planet than there's humans. And there are even more viruses out there, and there's no estimations on how many viruses there are. Well, they are called microorganisms, obviously, because they are small. This picture gives you an idea. This is a grain of sand. This is a cut through of a hair. A red blood cell, this is a bacterium. And a virus is generally about 1,000 times smaller even, so rather small. And this gives you, this is actually supposed to be a logarithmic scale here. This is a streptococcus. This is a bacteria that is found in our throat, for example. And some of the other bacteria, they are usually in this order of magnitude, so about 0.1 to 1 to 10 micrometers. And the viruses are a lot smaller. Now, what do I want to discuss with you today? Just give you an overview of the different kinds of microorganisms and then zoom in into the bacteria. And for the bacteria, we're going to discuss about the commensals, so our permanent inhabitants, those microorganisms that help us, so the good guys. And then we switch afterwards to the bad guys, so the bacteria that cause infections. And finally, we're going to look at the treatment of the infections by antibiotics. So again, if we want to look at microorganisms, we generally have to, we look at how we can, well, we consider how we can look at them. 0.2 millimeters is about the range until what we can see with the naked eye. We can go about 1,000 times more sensitive with a simple light microscope, and even more sensitive with an electron microscope. And you see here the different types of microorganisms, protozoa, yeast, bacteria. We can see those with the optical microscope, and the viruses we need an electron microscope to be able to see them. Now, the viruses I will not mention any further because I'm going to tell you more about that on Monday. However, if we look at the rest of them, they can be divided. We have some eukaryotes, and most of them, the bacteria, are the prokaryotes. So the eukaryotes that are there are basically the fungi and the parasites, and the parasites can be divided in the protozoa and the helminths, or the worms. Now, if we start with those, basically the biggest microorganisms, you see here some examples. Now, this is not a human worm. This is just a worm that you find in the soil when it rains, that the birds eat. But this is one that can be found mainly in the gut of small children. The eggs are usually found in sandboxes, for example, and the small children have these. They're not very dangerous, but they're annoying. And the same goes for this one. And you can see the size, so obviously we don't need a microscope to see these. The smaller parasites, the protozoa, are unicellular, and so we need a microscope, as you can see by the size. This one we had already discussed. I think you had it in skills lab and we discussed it briefly in the case. So this is a giardia. This is a parasite that is found mainly in contaminated water. It's not very dangerous. It can be treated. However, what you get is sort of intermittent cases of diarrhea. Now this one is a bit of a different story here. This is a plasmodium parasite that you can see here in these infected cells. It causes malaria. A lot harder to treat actually. And of course there's many more of them. Now the fungi can be divided in two groups. Oh, I'm sorry. I see that I put some Dutch words in here. So this is yeast. Sorry about that, yeast. An example of this is Candida albicans. This is a unicellular and it divides by budding. And this is the molds, and a good example of that is Aspergillus. Just to show you two pictures of those. This is a picture of Candida albicans, and what you see here in the cutout is a yeast cell, and a small one is budding out of it. And this is the way that they divide. So you see that happening also here, and for example here, and here are some bigger ones. So then basically they bud out, they are bound off and separate, and this is how new yeasts are formed. Now for the molds, this is something that, I don't know if you like this, but that is common, well, liked by some people. This is a mold, and you see that often they are multicellular. You see here the cellular structure. Basically this is the root, and what you see here on the top are the spores that are being formed. And so this is of the Penicillium family, in this case the one that we used to make Roquefort cheese with. And all these spores, they can fall off and they can go to other places and grow there again. And that gives you an idea why it's so hard to get rid of fungus, because they spread so easily. The spores are very resistant to heat, and they can spread to other places and continue living. Now, generally, so far in medicine you have only studied eukaryotes, and today we're going to focus mainly on the bacteria, and they are prokaryotes. So what are the biggest differences? Now, I don't need to tell you much about this, because that you should already know. But what is then different about the prokaryotes? Well, there's quite some differences in the cell wall. They don't have a nucleus membrane, so the DNA is free in the cytoplasm. They have structures outside, like a flagellum, for example, and that they can use to swim around. And also the ribosomes are different. And for the rest, there are some things that eukaryotes have that prokaryotes do not. So I summarized that here. When you look at the outer cell envelope, or in the case of the prokaryotes, the cell wall, they contain generally a lot of peptidoglycan. In the gram-negative bacteria, this is supplemented with lipopolysaccharides. And they generally have some structures on the outside, which eukaryotic cells do not. And they could be long sort of tails, like flagella, or they can be smaller hairs, like pili or fimbriae. And we'll come back to those a bit later. Now, mitochondria, lysosomes, and the nuclear membrane prokaryotes do not have, and the ribosomes are different. So, whereas eukaryotes have 80S ribosomes, the prokaryotes have 70S ribosomes. So this is a nice picture of bacteria. This is in fact an E. coli, and you see here that it has nice flagella, and it has also some fimbriae, or little hairs growing, and that it used for motility and adherence. And here you see, if we look at the bacteria, a bit more detailed structure, and this is divided actually in two sides, the gram-positive side and the gram-negative side. And again, you see here the free-laying genome in the middle. What we also see is the ribosomes. We see the structures on the outside, the pili. We see the flagella here. Some of the porins, so the little holes that it can use to extrude things to the outside. And in some cases there is a big capsule on the outside. Now let's look at these different things a bit more in detail. So the genome of a bacteria, just like ours, consists of double-stranded DNA. In this case, it's circular, and generally you should think of it like an elastic band. So it's an elastic band, and you just completely twist it up, and you roll it small, and it becomes so it's completely circular but folded. Then you have the bacteria might contain plasmids. They don't always have plasmids, but when they have plasmids, these are also DNA or circular DNA fragments, and they are generally non-essential. So what that means is these are nice little extras that the bacteria can have, and generally they contain interesting things like antibiotic resistance or virulence, or things that could give the bacteria an edge when they want to infect. Now, if we then look on the outer cell wall structures, generally they contain lipoteichoin acid, and this is used for adherence. Now, adherence I will come back to during this lecture, because this is an essential step in causing an infection. Another structure is lipopolysaccharides. This is mainly found in gram-negative bacteria. They are also called endotoxins, because in humans, when you have a lot of lipopolysaccharides, or which is also called LPS, or abbreviated as LPS, this can cause inflammation and could lead to shock. But I mean, although they are a toxin for us, they are just a cell wall component for the bacteria. And then the ribosomes, like I said, it's 70S ribosomes consisting of a 30 and 50S subunit. The bacteria might or might not have a flagellum that is used for, so this is the biggest tail, and this is used for motility. It might or might not also have pili or fimbriates. These are the smaller hairs or little tubes, and they can be used for two different things. Well, they can also be a little bit be used for motility, but mainly for adherence and attachment. But also we speak of sex pili because these can form tubes between bacteria, and through these tubes mobile genetic pieces like plasmids can be exchanged. And we will come back to this process later. And then also, this is an optional feature. Bacteria might contain capsules. And with a capsule, what that means is these are usually polysaccharides, and the polysaccharides are being extruded by the bacterium. And we just discussed in the last few weeks the immunology, and you know now that if your immune system wants to fight a bacterium, either phagocytes or antibodies, they need to attach to the bacterial cell, or complement, for example. They need to attach. But if on the outside of the bacterium there's a whole layer of slime, basically, then the phagocytes and the antibodies, they cannot bind. And in this way, it protects against phagocytosis. So this is a sort of a virulence mechanism, an escape route for the bacteria to keep itself safe. Also, you can imagine that if the bacterium wants to attach to human cells, and you have a nice big slime layer, that can help it stick more to the surface. So it can also be used to facilitate attachment and adherence. Now, bacterial growth, I mean, we speak about a lot of different bacteria. And I mean, they don't always all grow the same, but on average, so for most bacteria, under good conditions, they grow and they divide in about 20 minutes. So how does this happen? You see here a picture of a bacterial cell. What you see is that the genome is being duplicated, and you basically see two little balls. So the genome is being duplicated and then pulled to two sides. That you see here, and then a septum is being formed in the middle, and the septum is being put all the way through, and then basically what happens is that two different cells are being formed. And you see here in a microscopic picture of a bacillus, and you see the formation here of this septum, this cross wall in between them, and you see already that the two pieces of genome, they are already separated to the two sides. So there are thousands of different species of bacteria, and they have very different. They have different shapes, they are in different arrangements, they have a different gram color, they metabolize in different ways, they are have different functionalities. Some of them are pathogenic and others are not. Some can live, for example, on plants and others do not. Some can form spores and others do not. And I want to zoom in a little bit with you on these differences. And here you see a picture that nicely demonstrates the different shapes and arrangements. So what you see here on the left are the cocci, and cocci are basically sphere forms. And these sphere forms, these spheres can be single, so just loose cocci, but they can also be in groups, and they could be with 2 together, or with 4 together, or nicely and neatly stacked like a cube. Or some of them, like the streptococci in our throat, they like to form long chains of cocci. And these are staphylococci that we will come back to quite a lot later. And they are actually cocci that are in little grape bunches. So they are not in the neatly arranged groups, but they are in shapes together, in clusters together. So based on the shape, so the cocci form and the arrangement, we can already have quite a good idea which microorganism we are dealing with. And the same goes for the rods or bacilli. And you have fat rods and thin rods, and rods that have basically like a ball on the end, and some of them that are a bit bent, like the Vibrio cholera, for example, or that are a bit spiral shaped, like a Campylobacter. So also here, I mean, we can have a lot of different ones, and by looking at them under the microscope with people who have experience with this, they can have a good idea of what we're dealing with. Now then we have the gram staining. It was developed at the end of the 19th century by a Danish Dr. Gram. And basically what is easy to remember is positive is purple, so PP. And the negative is the other color. And usually the dye that we use for that is red or pink, but you can also use another color. So why is this purple stain on the gram positives? Here you see cut-throughs of the gram positive and the gram negative cell wall. And the essential thing is the peptidoglycan amount. So the purple strands here represent peptidoglycan layers, and you see that the gram positive ones have, thick layers of peptidoglycans. The gram negatives, however, have peptidoglycan, but just a small layer, and they have an extra layer of lipopolysaccharides, so the LPS, the endotoxin that I talked about before. And they have a pyroplasmic space in between, and usually there can be different enzymes, and usually enzymes that are not so good for antibiotics. So what happens when we do the gram staining, we use a purple dye, and the purple dye actually gets trapped in the peptidoglycan. Now, because there's a fat layer of peptidoglycan here, a lot of the dye gets trapped and the bacteria color purple. Now here, this is a very thin layer, and the dye just washes out again, and then you can counter stain it with another color, in this case pink or reddish, and then so the gram negatives are this color and the gram positives are that color. Now what you see here are actually staphylococci, and you see that they are little circles and they like to be in small clusters, and they are gram positive, so we know that these are staphylococci, and these could, for example, be E. coli. They are rods and they are gram negative. Now, not only based on their size and arrangement and gram color we can separate them or divide them, but also on what kind of atmosphere they like. And some microorganisms, they need oxygen to produce energy. An example of that is Pseudomonas. Some of them, they do not tolerate oxygen at all. They will die, like the Bacteriology. And some of them, they can live in both circumstances. Now, it has to be said that usually they prefer one or the other circumstance, but they can survive in both atmospheres. And E. coli is a good example of that. And that also explains why E. coli likes it so much in our GI tract, because there are pieces where there's more oxygen and there's pieces where there's almost no oxygen left anymore. Now, there's also some other ones, like, for example, microaerophilic, where you need a specific amount of oxygen, but you don't need to remember those. Now, some microorganisms can form spores, and basically what these spores are, and this is a bit different than the spores that fungi form, because in this case these are very thick-walled survival forms. And you see here a bacillus, and this bacillus inside is forming a spore, and to the green structure here, and eventually the cell around it